This is a support video for students or teachers alike engaging or interested in the microarray lesson. Now, this microarray lesson was previously described in the Nova version, so I recommend that if you haven't been introduced to this lesson yet, I would recommend going to the link right here, the microarray lesson plan, the Nova, which holds more truer and more precisely to the procedures of the microarray lesson. It also, clicking here, will give you access to an overview tutorial video, much like this one, but it gives a better description from beginning and closer to the end. This video leaves off a comparison between the two versions, the traditional Nova version and the newer Cabbage version, as it's called, as well as not digging into much detail about the final part of the lesson, which is discussing results as well as looking at possible medications to treat your patient with who's suffering from cancer. So in other words, this video will take off and add a little more detail where this Chapter 13 Microarray Lab Nova version video left off. So it picks up where it left off, right? And it gives you another way of looking at the lesson using a new pH indicator or your DNA sample, using a different substance for the DNA sample. So the other version is this CSCS lesson, Microarray Cabbage version. We're going to go ahead and click on it. And in general, it will have the same general approach as the other lab, same forms. This is the first indication that the lesson is different. You can see that the DNA sample looks a little different, looks like a blood sample, hence the Cabbage version. So they're related. And as we keep going, we find ourselves at this part right here. And this is where it's most different from the prior traditional Nova version. In the Nova version, you had 3 colors within each cell here, or well. Each of the wells from the microarray could be either light pink, clear, or dark pink. You can see the colors here are much different. You can have purple or blue. You can have green or red. These colors are far different from one another and reduces the chances of misidentifying the gene activity level of the gene. So let's compare the results from the Nova traditional and the newer version, the Cabbage version. Let's just look at and examine images of the results or gene activity for patient 2 if we were using the Nova traditional DNA or the DNA from the Cabbage version. In the Nova version, if a gene is overexpressed or producing lots of protein, more than average amounts of protein for a particular gene, it would be dark pink. If that gene was underexpressing protein, so it was a gene that's silenced or not making protein, it would show up as light pink for the well for a particular gene. And if it were functioning on an average typical level, producing a normal number of proteins, that gene would be identified as a clear color. So overexpressing is dark pink, underexpressing is light pink, and normal functioning gene activity is clear. So you can see this 0 right here, this well for this gene, the ERBB2 gene, would be normal, and it would be clear inside. If it were underexpressed or inactive, if it was a gene that was silenced, like this gene here, we would notice it's light pink. And in a cell or well here, for the BCL2 gene, it should be dark pink. Let's look at the real results of what that looks like. So here's a typical results, what you might expect when doing the Nova. And you can see the DNA has been applied, so the DNA sample was applied to each well or each gene in the microarray. But just look at this, how close clear and pink can be, or the various degrees of pink you can have. This pink is darker than this pink. Is this pink dark enough to be considered a highly active gene, or must it be this dark to be highly active? Now, while some might argue that yes, you can identify this clearly as normal functioning, silenced gene, highly active gene, silenced gene, normal functioning, silenced, normal functioning, etc. Others, and particularly students, often, even college-level students, when doing this lab, can confuse easily what is light pink or silenced genes, which genes are clear or normal functioning, and which genes are dark pink or highly active. So that gave birth to the new lesson, which is using the cabbage color. So here's the traditional NOVA version results, where DNA has been applied from your patient. This would be patient number 2. So patient number 2's DNA has been applied to each well or gene, and you can see various colors. The degree of difference between if a gene is, say, silenced, light pink, and if it's normal functioning is pretty close. There's not strong differences. So you really have to be a very careful observer to identify which genes are silenced or inactive, like this, versus normal functioning, like this. And it's common for high school students or even college students when doing this microarray lab to misidentify which genes are active or inactive. So again, identifying whether a gene is highly active or making large amounts of protein, the darker pink is somewhat easy to identify, but the difference between the silenced or inactive genes versus the normal functioning genes is rather close. So let's look at the other option, which is the cabbage version, which is what this video is made for. And you can see clearly there are strong differences between identifying a gene as highly active, inactive, or normal functioning. So going back to the page where you do the lab, we can see the key, so it's just explained so we don't get confused. If the gene is overactive or producing high levels of protein, more than normal, you would see a blue or purple color. If the gene is silenced or inactive, it's not making any protein, you'd see the green color, and then red color means a normal functioning gene producing average number or amount of protein. So knowing this key and knowing that overactive is purple or blue, inactive or silenced is green, and normal average functioning gene is red, let's go back to it and just see just how easy it is to identify the difference. And here you have normal functioning genes producing normal amounts of protein, and you have your green silenced genes, and you have your overactive or producing higher than normal amounts of protein as purple or blue. Now you can see here there is a little variance, the blue and the purple here. They're of the same activity level. They're producing high levels of protein. So you got to be really watchful and make sure that you're paying close attention still to the colors, because just based on what residue might be remaining in the tray or contaminants can still affect how easy it is to identify gene activity level. But at least when you're comparing it to the normal functioning gene in that particular well, it's still a pretty clear difference. So using the cabbage version potentially might be easier for students to identify which genes are active, inactive, or normal functioning. And this data, of course, will then be used for the next step of the lab, which is reporting on which genes are active, inactive, and normal functioning. And it gives you an opportunity to see and test really if the cabbage version gives more clear results. So you'll have multiple groups looking and investigating patient number 1. You'll have multiple groups investigating patient number 2. And it gives you an opportunity in the data sheet, so the results that when the students enter it, just to see how closely similar their findings are, what they're reporting, their data, right? Their data on each patient and their gene activity. All right, so as you go down, you see the next part of this is the data form. When you click on the data form, it opens up a spreadsheet. You can see prior students doing the lab's results here. So we have here individuals who've clicked on the data form so that they can report on which genes they found for each patient as being active, overactive you could say, turned off or silenced, or normal functioning. Just case in point, when doing, and these are the results for the traditional NOVA, what you get is, one example might be this right here. So both of these teams investigated Jason Derulo as their patient. One group identified his BCL2 gene as on, while this group did not. Now, this is the beginning of a good discussion on quality data, making sense of data. This is a great opportunity potentially to redo the experiment using realistic results for data when you have conflicting information or data, right? And you can use this as a teachable moment and to just practice really good data collecting skills and to discuss potential false positives or falsely identifying a gene's activity level. That also can take time. Redoing a lab and allowing students to argue is helpful and develop skills for everyone, especially if you're doing the lab. It's great. You're engaging in scientific practices. But if you're short on time or you want to make sure you have more clear results for students, then the Cabbage lab should avoid these false positives and see a little more commonality and clearer results for different groups doing the same patient. Anyways, but you can see here the data sheet. You enter in your data for gene activities for high level activity or high production of protein, low production of protein, and normal. Once that information is entered in, that is the beginning of the class discussion, and you can start finding patterns of for which genes are on, off, and normal. And we can really start learning that genes overall aren't always on, they're not always off. They may not be making protein. And that really could be the foundation for just understanding certain disease like cancer in this example. So let's go, what would be the next step? So let's look at the lab and kind of move forward and see. Once you've entered in your data, the next part really is identifying your patient needs. As far as you being their doctor, what would you advise for them as far as medications? And let's see if we can zoom in. So you can find out by investigating, here's an example we'll discuss, but you have 7 different medications and you want to be able to advocate and identify the most ideal medication or prescription for your patient suffering from cancer. So the example shows you here, if you were thinking about considering prescribing zaclofloxacin, you wouldn't be able to, or you're not supposed to use this medication if your patient has certain genes active, the BCL2 gene or the DPYD gene. If the gene is active in the patient, apparently that can cause conflicts. So apparently if that gene is active and producing proteins, this medication will interact with that protein that's being produced from those genes, and that medication and the proteins combined cause some kind of problem for the patient. Side effects, difficulties, it could be fatal or just uncomfortable. Either way, if the genes are active and making proteins, the medicine doesn't mix well or react well with that protein in the body. If they are positive, you cannot recommend it for patient 1 or 2. You have to find the gene activity level for your patient for each of these and come up with a treatment plan for your patient. Once you've identified which prescriptions are most appropriate for your patient based on what genes you've identified as active, inactive, or normal functioning, the next step is to report on that. All right, so you now know what prescriptions you feel are ideal or most appropriate for your patient number 1 or 2. So you go to the post-lab analysis, and this gives you your final opportunity to enter in information. So you put your name, and each individual will submit their own analysis and report. You can see you'd answer, based on the genes active, provide important trends or results that should be reported. So what did you notice as far as the genes active, normal, or silenced? What does that say about your patient and what's causing their cancer? And finally, you report on what medications you think are best for your patient, and you go over the spreadsheet to generate some discussion with your patients, as you can see here.